A heart that develops cancer starts out just the same as any normal heart, but any number of cells are hijacked or mutated so that they begin to grow uncontrollably. Whereas the cells here have stopped growing and are only going to grow to repair any injuries to the heart, in a cancerous heart, cells will grow out of control. So there's some cells that grew where they weren't supposed to, and they're going to keep on growing until they form what is called a tumor. A tumor is simply a growth of cancerous cells. And these tumors can become so large that they impede the function of a critical organ. So here, this could impede the function of the heart. And as I'm sure you can imagine, that's very detrimental to the patient's outcome. Let's look a bit deeper about how cancer tumors work. The first thing to note is that there's no one single type of cancer cell. Any kind of cell can become cancerous when it is hijacked and mutated so that it keeps on growing. Cancer tumors are usually made of a large variety of cells, and all of these cells serve different functions and are essential for making the cancer tumor grow and spread. So let's say we have a cancer tumor here on the audience's left, and we have some healthy skin cells in a grid on the right. Now, the way that cancer spreads is because this tumor is so large, due to gravity, part of the tumor is going to fall off, drop off, and move through the bloodstream until it happens to land on some healthy cells. From there, because it's a cancerous cell, it's going to grow out of control, and as it gets bigger and bigger, it's going to starve and destroy a bunch of healthy skin cells. And eventually, we have another large tumor and non-functioning skin. This is how cancer spreads, so that even cancer that starts in a very innocuous and unimportant part of the body can reach critical organs and kill a patient. And it's important to note that this little cell growth isn't a trivial problem. According to the GlobalCan estimates for cancer around the world, there were 19.3 million new cancer cases in 2020. That's a huge number. And perhaps even more saddeningly, there have been estimated to be 10 million cancer-related deaths in 2020. It's estimated that cancer accounts for 1 in 6 of all deaths across the world. That's crazy. 1 in 6 deaths to one type of disease. One of the reasons that I care so much about oncology and studying cancer is because I experienced having a family member, someone who was really close to me, pass away due to cancer last winter. My grandmother, who was a pharmacist and who actually inspired me to get into science in the first place, passed away due to kidney cancer. And each of these 10 million people leaves behind a family and people that are not going to be the same without them gone. Cancer is hugely impactful on the world for all of the wrong reasons, and we need to do something about it. So, back to those tumors. What makes them quite so deadly? Well, one of the reasons they're so deadly is something called a cancer stem cell. Every tumor has a couple cancer stem cells in it. I've highlighted 2 of them in red in this diagram. And so the interesting property of cancer stem cells is that they can shift and they can mutate their form. So for example, this hypothetical cancer stem cell currently looks like a circle. But if it so chooses, it can shift itself into an oval cell. In this way, it can change its properties so that it serves whatever need the tumor has. So if the tumor needs to rapidly grow, it will change itself so that it can rapidly spread. If the tumor needs to hide from some immunotherapy, it can camouflage itself, so the immunotherapy can't find it. And that's one of the reasons why cancer treatments struggle to fully eliminate tumors. So there are 3 main types of cancer treatment. The first is surgery. It's effective against single tumors that you can just surgically remove. However, it is highly invasive, and once the cancer has spread or metastasized, as I show in a previous slide, the surgery doesn't work because you can't remove someone's entire circulatory system via surgery. That's called killing them. Chemotherapy is another kind of cancer treatment. It basically uses very powerful chemicals that can kill all of the cells they come across in your body. And while they're very good at eliminating cancer cells, they're indiscriminate killers. They inflict huge collateral damage, and the side effects are so bad that they can even cause new cancers and be worse than the cancer itself. In the end, the side effects to chemotherapy is more than likely what ruined my grandmother's life, more than the cancer itself. Immunotherapy is the newest and some would argue the most promising kind of treatment for cancer, and that's what I'm going to be focusing on today. It is much more discriminate. It uses compounds that are less powerful than chemotherapy, but they can accurately distinguish between cancer cells and non-cancer cells. However, we don't have immunotherapies for all cancers, and they're susceptible for being tricked by cancer stem cells. Because again, if a cancer stem cell disguises itself as a healthy cell, then the immunotherapy will have no way to tell. So let's illustrate some of the problems with immunotherapy. Here I have 4 balloons, and I have a needle. The needle represents the immunotherapy, and the 4 balloons represent cells in your body. Let's say that one of these balloons is a cancer stem cell. Can anyone tell me which one? Yeah, Madison, go ahead.
Is it the third one?
I couldn't even tell you because they're all the same. I'm sorry, almost all of the interactives I have are going to be rigged against you, just a PSA. So, what do? We could just attack the third one blindly, as Madison suggests. An experienced oncologist may say that that's a very bad idea because you don't know if you're going to actually get the tumor and you're basing it off of no evidence. One thing that you could do is you could just pop all of the balloons. If we really need to get rid of his cancer stem cell, we can just do this.
Well, you can go one at a time, and then like you can see.
Well, if you can't tell if there's any cancer stem cells remaining though, until after they grow again. So you have to pop all of them. And see, we got the cancer stem cell. They're all gone. However, we've also killed 3 very healthy cells. This is not a good ratio of success, and this inflicts huge collateral damage. The whole point of immunotherapy is not to inflict collateral damage, so this is not good. This is not an effective solution. All right, I'll make it a little bit easier for you to tell which balloon the cancer stem cell was through a label. Now, can anyone tell me which balloon the cancer stem cell was? I don't believe this. All right, so as I've very clearly illustrated, you had no way of knowing that it was the third balloon. And that was indeed the cancer stem cell. But now that there's a label, it's much easier to tell. And as a result, the immunotherapy can pop the correct balloon, all of the healthy balloons are still there, and everyone is happy. Yay! This is exciting. Except there's a problem, which is that I just hypothetically introduced a label that was not there. And this is exactly what researchers tried to do in the paper I'm presenting on. They asked the question, how do we label tumors and cancer stem cells, making it easier for immunotherapies to identify them? Before we continue, does anyone have any ideas on how this might be able to be done? I had no ideas. Yes?
Well, cancer cells multiply more, so maybe something where the more you multiply, the more visible it is.
Yeah, that's a good idea. Cancer cells do grow much faster than normal cells, and a lot of the time that is how they're identified. One of the difficulties with that solution, though, is if there's even one or just a couple cancer cells that are left behind, then they'll just grow again, and since not all cancer cells are growing at any one moment, it can be hard to tell. Yep.
I'm not sure, but if there are any proteins or chemicals like hormones that are associated with really fast growth of cancer cells, then you just make it target that?
Yeah, that's a great idea, targeting the cells that are most deadly in the cancer tumor, maybe even a cancer stem cell, targeting it and destroying it. Aura.
I mean, if there are any specific, like, sort of like you said, molecular factors that can be identified that distinguish cancer cells or cancer stem cells, that's something that will react to those cells.
Yeah, absolutely. I think that if you combine all 3 of your ideas, you really did get to what the researchers did here. So the paper I analyzed is titled LGR5 marks targetable tumor-initiating cells in mouse liver cancer, or put more simply, something called LGR5 marks cancer stem cells in liver cancer. So that's exactly what they did. They found a compound that exists around cancer stem cells and targeted it. So what is that compound? What is LGR5? Well, LGR5 is naturally found in our nervous system. It's not actually found in our liver, and it is a rhodopsin-like protein structure, but that's not relevant, and you don't need to know what that means for the purposes of this paper. All that you need to know is that it has been previously shown that LGR5 likes to hang out near cancer stem cells. So you'll find more of it where cancer stem cells are than you normally would. In this way, it acts as a label. And what the researchers are hypothesizing is that you could use this label to help guide an immunotherapy to be able to eliminate a tumor. And as you can see in this diagram, it looks like the immunotherapy has been pretty successful. There's only one cancer stem cell left. But what happens if that cancer stem cell changes shape, say from a circle to a cloud cell? Now what? Yeah, exactly. Normally, the immunotherapy would be really confused, and this is a lot of the ways that immunotherapy actually misses cancer stem cells, is because they change like this. However, because the LGR5 is still there, it can act as a label for the cancer stem cell and allow the immunotherapy to hit its target and eliminate the tumor. Now, if this actually worked, This would definitely improve patient outcomes because it would make existing immunotherapy much more effective. So the researchers decided to test their hypothesis. Can LGR5 be used to target cancer stem cells and tumors, acting as a label? And would that actually make immunotherapy more effective? So the first question the researchers had to answer was to check if LGR5 was accurately labeling the liver tumors, so if they were actually finding LGR in the right places. So if you would please turn your attention to figure C on your left, you'll see that there are 3 columns on the x-axis. The first one is normal. That signifies a normal liver, no cancer, nothing wrong with it. The second column, labeled injury, symbolizes an injured liver. So the liver has been punctured or hurt in some way, but it doesn't have cancer. And the rightmost column, tumor, indicates that a liver cancer tumor has been found on the liver. So there is cancer present. The y-axis represents the percentage of LGR-expressing cells that are present. So the higher up a point is on the y-axis, the more LGR is there. And as you can see, there is far more LGR in the tumor than in the normal or injured liver by a huge order of magnitude, from 0.5% to over 10%. This supports the idea that LGR5 is working great as a label. Jonathan, question?
Yeah, how did they find patients to have determine the concentration?
They used mice.
Oh my.
Yeah, I was going to talk about this in a moment here, but they used mice in order to carry out this study, because really putting active liver cancer that has a very terrible prognosis rate into a human wouldn't be ethical. The survivability rate for liver cancer that has spread is around 2% after 5 years. It is really deadly. It's tragically common, and even if it's detected early, the survivability rate is around 15%. So it's a serious problem. But as you can see, the LGR5 is there where there is cancer, where there is a tumor. And when there is no cancer, it is not there at all. When it's injured, it's barely there. This ratio is much better than the ratio we got by popping 3 incorrect balloons and 1 correct balloon. So this label is definitely helping. If you look at figure D on the right, this shows a very similar claim. Again, on the x-axis, there's 2 columns. The rightmost column is a tumor, so where cancer is present. The leftmost column is the area immediately around the tumor. So there's no cancer, but it's near cancer. And once again, as you can see, there is far more LGR expressed at the tumor than in the area around it. And taken together, this supports the fact that LGR5 is working as an accurate label for cancer stem cells and liver tumors. Question?
Yeah, so if we're using the LGR5, it was earlier said that it's common in the nervous system. So how do we know that the immunotherapy will be targeted solely in the liver and not go off the spinal cord or something?
Yeah, that's a great question. So in order to actually add LGR5, the researchers just took it from where it normally is in the nervous system and from outside the mouse, and then put it into the mouse's liver. And then the measurements were all taken in the liver. And so as you're going to see on the next couple slides, when they induced the drug, because the cancer is only in the liver, what the researchers did was they added anti-cancer treatments to this experiment. And since those cancer treatments were only added to the liver, they probably were very unlikely to spread through the nervous system. But that is something interesting to consider, is that there is lots of LGR in the nervous system, and so as a result, it probably wouldn't work as a label there, because it's everywhere. So the next step for this experiment was to figure out whether these labels actually help patients. Because having the label is one thing, but LGR5 doesn't do anything to cancer. It kind of just says, hey, there's cancer here, better watch out, and then just stays there until it is eventually killed or washes away. So it's not an effective treatment. And so what the researchers did was, after adding some liver cancer tumors into a mouse, again, this is the part which wouldn't have been ethical in humans. So after adding liver cancer tumors to a mouse, they administered 2 anti-cancer treatments. The first one is called 5-fluorouracil, or 5-FU. And in this compound, what it essentially does is it has an affinity for LGR5. So this compound will seek out LGR5, and it has been shown to have some positive effects against liver cancer. So they're essentially using 5-fluorouracil to direct the anti-cancer treatment to the labels. The second compound they added is DT, which stands for diphtheria toxin. And diphtheria toxin is a toxin, which just means that it kills the cells that are around it. And so it has also been shown to decrease the size of cancer treatments. And it's sort of a chemotherapy-based treatment for cancer because it indiscriminately kills. The researchers proposed that by doing this, the labels would allow the combination of 5-fluorouracil and diphtheria toxin treatment to remove the tumors in the mouse accurately, and the mouse would survive. So did this work? Yes. The LGR5 label did, in fact, make immunotherapies more effective, and here's how. Here on this figure, the x-axis is time since the tumor was transplanted into the mouse, and the y-axis is the size of the tumor. Again, the bigger the tumor, the more likely it spreads, and the worse patient outcomes. So the higher up a point is on this graph, the worse. The yellow line, which you see here at the bottom, that represents the combined treatment of 5-fluorouracil and diphtheria toxin. And as you can see, after the treatment was administered on day 6 of the tumor's growth, the tumors hardly grew. The tumor growth was stopped, and this is exactly what immunotherapy is supposed to do. And so this combined treatment with the labels was highly effective. But not only that, it was far more effective than any of the controls. The black line on top shows the liver tumor not being treated and just being ignored, and as you can see, it grew massively to around 50 millimeters cubed. That's a huge volume. That's enough to metastasize and likely kill a patient. And the green and the red lines represent 5-fluorouracil and diphtheria toxin being used as treatments on their own. And again, neither of these are as effective as the combined treatment of 5-fluorouracil and diphtheria toxin with labels. The labels have made the treatment significantly more effective by a large scale factor. Taken together, this data means that the labels have made an existing immunotherapy treatment more effective and improved the patient outcomes of these mice. Now, this is great, but what does it mean for patients and what does it mean for the broader field of cancer? Well, this paper has shown that we can make markers of cancer stem cells and label tumors, and this allows more compounds to effectively and precisely target tumors. This makes immunotherapy, which is already an area of great promise, work better. Not only does this decrease the side effects of immunotherapy drugs because it decreases collateral damage done, but it also accelerates and simplifies the drug discovery process. It's much easier to create a compound that can find a label than to create a compound that can sniff out a cancer stem cell. So the researchers' hope, and my hope as well, is that not only will the development of labels like LGR5 lead to more cancer drugs being discovered faster, it will lead to each cancer drug having less imposing side effects, and it will lead to the cancer drugs more effectively being able to get rid of tumors and not leave any cancer stem cells remaining, improving patient outcomes.